Hi, I'm Jonah Comstock, editor-in-chief of pharmaphorum. I'm here on day 4 of ASCO. That's technically the second to last day, but it's the last day for me. And I'm joined by Luke Piggot, principal scientist at Debiopharm. Um, Luke, thanks for joining with me.
Yeah, thanks for having me.
So you guys don't have a ton of stuff here at ASCO, but you presented a lot at AACR about a month ago, and you also have some recent exciting news from the FDA, right?
Yeah, absolutely. At AACR, we reported the first clinical data for our combination of PKMYT1 inhibitor lunresertib together with zedoresertib, which is a WEE1 inhibitor. So it's a novel combination that demonstrate really strong synthetic lethality. It was the disclosure of our phase 1 dose escalation results, and we're seeing a really positive response rate there of about 60%. And on the basis of that data, we also received fast track designation from the FDA.
Which means you'll be able to move much more quickly to get this product into market.
Absolutely. So we can leverage that opportunity to have more regular interactions with the FDA and also get some more in-depth guidance on the best way to develop this, which is very useful for us, given that it's a novel combination as well. Neither drug is approved in its own right, so it's a complicated development pathway.
So one of your areas of expertise and one of Debiopharm's focuses right now is the DNA damage response. I got to say, I'm not super familiar. So talk to me about what that is and how it's changing the paradigm here.
Yeah, absolutely. So cancer cells essentially carry a lot of genomic instability. That inherently induces some DNA damage within the cells, which would normally lead to the cells dying. But it's actually a component of one of the hallmarks of cancer cells that allows them to grow uncontrollably. So in terms of DNA damage response, if you prevent those cells from being able to repair that DNA damage, then actually it will drive them into a natural cell death instead. So what we're focusing on is really alleviating some of those checkpoints that would normally allow the DNA damage to be repaired, and instead driving them into a cell death.
So this is kind of a different way to attack tumors.
Yes, exactly. It's a little bit paradoxical when you think about it, in fact, because normally you don't want to drive the cells into further around their cell cycle. But in the instance where it's carrying a heavy amount of either replication stress or underlying DNA damage, it actually leads to the cell not being able to cope with that and then leading to cell death.
And is this an issue where making sure that this targets tumor cells but not healthy cells is a problem, or is that kind of built into how it's?
Yeah, so all cells rely on this sort of capability of being able to repair their DNA damage. Now, tumor cells, as a virtue of the fact that they carry this high genomic instability, are far more reliant on it than normal cells. So that's what creates the therapeutic index there, so that you're having a greater effect on the tumor cells than you are on the normal cells.
Talk to me about sort of overall strategy at Debiopharm. What are your kind of beyond what we just talked about? What are your focuses in mechanism of action? What are your focuses in therapeutic areas?
Yeah, so we are still interested. We've got the two drugs, obviously, in the DNA damage response sort of area, but we are also heavily invested in progressing different forms of ADCs. We look at various different formats of ADCs as well, bispecifics, monospecifics, but also we're doing some work with some innovative payloads in that space. And we use our proprietary linker technology as well, which is a very sort of stable linker product. And we believe that that's going to bring a number of advantages over the existing ADC technologies that are out there today.
Tell me a little bit more about that, because obviously with ADCs, you've got the target and the payload, but then the linker is sort of that third piece that people don't always talk about as much. Why is that important, and what advantages does your technology bring?
Yeah, absolutely. So I think, yes, the target and the payload are typically the focal points, but at the end of the day, the sort of the key piece that links it all together is the linker, right? And so having more favorable properties around the linker technology actually, we believe, brings a lot of benefits and actually can be very differentiating. So things like stability is a critical element for toxicity. So the vast majority of the toxicity in ADCs is driven by the payloads. And so unfavorable release of your payload in the systemic sort of system can lead to toxicities, which are then obviously unfavorable for patients. And then it gets less of the payload to the tumors. So again, that then translates into a greater efficacy if you can stably take those to the tumors. There's also a whole number of different dynamics around whether or not it's cleaved in the tumor microenvironment within the cell, how well it cleaves, how quick it cleaves, and all of those things can have an impact on the efficacy of our products.
Got it. So the thing that makes ADCs work, the targeted nature, the linker just makes that work better.
Exactly. So the linker technology is responsible for making sure the whole system works efficiently, basically. Yeah.
And we're getting pretty far into the conference. So tell me what are some things you've seen and heard so far that have been interesting to you?
So I think two things I would highlight, and they were both happened to be yesterday actually, was the disclosure of the PD-1 VEGF bispecific antibodies, and it was a readout from a trial that was ongoing in China, showing some positive results in lung cancer. And I think it'll be really interesting to see how that translates into the global population. It's showing some initial early signs that this could be a promising approach. But I still think there remains a lot of question marks around how well these data sets translate from the Chinese run trials into the global population. And then I think the other thing that I would highlight is the disclosure of the KRAS data from Revolution Medicine. That particular study in pancreatic cancer is a categorical leap forward for those patients, and it's really encouraging to see those patients get something new.
Yeah, a lot of buzz about that KRAS study. But also, I'm glad you mentioned, you're talking about Harmony 6, right?
Yes.
Yeah, I think you kind of put that in the larger context of this increasing role the Chinese biotechs are playing in the ecosystem, and that question of sort of will the data translate? How long do you think it'll take? How much data do you think it'll take before we sort of start to have an answer for that question? We can really know sort of what we should think about studies like this.
I mean, there's no question that they're doing these studies remarkably quickly. And I think that that's, something that can be leveraged as a sort of a global approach as well. I think there's a lot of things that both sides can learn from each other as well. And I think that the most efficient thing to do would be to have a more harmonized approach, taking the best learnings from both sides of that story, right? And so that's where I think we'll end up heading is, these Chinese developments are going to want to be more translationally relevant to the global approach. And I think that'll come as a consequence of working more and more across those borders. I think it's still going to be a little while. We probably need to see a few more of these phase 3s and even some of the phase 1 studies read out before we can categorically say, here are where the discrepancies are going to lie, and this is the best way to try and address those differences. But I think within the next few years, again, given the speed that the Chinese are working at in that area, we should have a much better view of all of that.
So what's next for Debiopharm? Any more data on the horizon? Anything you're excited about at upcoming conferences?
Yeah, I mean, I think for us, we're going to disclose some more data at ESMO with the WEE1 inhibitor, so zedoresertib. And then I think that the next sort of big landmark time point for us would be starting our phase 2 clinical trial for this combination with the PKMYT1 and the WEE1. Now that we have the proof of concept data in the ovarian cancer patients, we're looking at expanding that to more indications and initiating the phase 2 clinical study. And hopefully that'll come sometime next year as well.
Great. So it's an exciting time for you.
I think so, yeah. We're very excited about the data that we've generated so far and the benefit that it's bringing to the patients so far as well.
Great. Thanks so much for chatting with me.
Thank you.